Hello, and thank you so much for tuning into my presentation entitled Feather Corticosterone Variation and its Phenotypic Correlations in Purebred Domestic Turkey Lines. My name is Naim, and I'm studying zoology at the University of Guelph. This summer, I've been working with Professor Christine Baes and PhD student Emily Leishman in the Department of Animal Biosciences, studying corticosterone levels in turkey feathers. For some background information on the topic, corticosterone is the main stress hormone in birds and is deposited in feathers during growth. This hormone can indicate an animal's welfare and may ultimately affect reproduction, productivity, and fitness. Emily's previous pilot study showed that feather corticosterone can successfully be measured with an ELISA kit, and cort levels vary between individuals and genetic lines. Emily found that turkeys selected for growth traits had significantly higher feather corticosterone than those selected for reproductive purposes. This variation allows us to further investigate feather cort as a phenotype. My personal objective this summer was to gain understanding on the topic of feather cort and avian welfare to prepare for the independent research project I will begin with Dr. Baes in the fall. My main goal in the lab was to prepare a large number of feather samples for analysis to observe the changes in feather cort. Moving on to methodology, 865 feathers were gathered at the abattoir from purebred male turkeys. 3 different genetic lines were used for this study, which we will refer to as line A and B selected for reproductive traits and line C selected for growth traits. All birds were raised in identical husbandry conditions. All individuals were genotyped and have available production and meat quality data. The steps involved in measuring feather cort include cutting up primary feathers into pieces less than 5 millimeters squared. Feather bits were then mechanically homogenized into fine powder using the bead blaster. The next step was to weigh out the feather powder, add methanol to each sample, and incubate overnight at 50 degrees Celsius. This allowed us to extract the corticosterone from the feather powder. Vacuum filtration was used to filter out feather particles from the methanol. The methanol was evaporated under nitrogen gas at 40 degrees Celsius to leave us with the residue of steroid hormones. Dried samples were then reconstituted with an assay buffer, and corticosterone was measured with an ELISA kit. In conclusion, because feather corticosterone varies between individuals and genetic lines, it has the potential to be used as a novel welfare phenotype in domestic turkey breeding schemes. But in order to verify that feather corticosterone will be a reliable and relevant phenotype, we need to perform further analyses, which I will assist with in the fall as part of my independent research. Future directions resulting from our research include 1. Comparing feather corticosterone levels between genetic lines with an even larger sample size to confirm line differences. 2. Performing feather validation of feather cort methodology, for example, biological validation comparing feather corticosterone between stressed and controlled birds. Does greater stress directly result in higher corticosterone levels? 3, exploring whether there is a phenotypic correlation between feather cort and other traits such as productivity, growth, meat quality, and walking score. If feather cort has no relationship to any relevant traits, then it might not be a very useful component for companies to include in their breeding strategies. 4, exploring the genetic basis of feather cort levels, including heritability and genetic correlations. Can feather cort levels be inherited from parents to offsprings? Can we identify genes associated with feather cort levels? That concludes my presentation for today. Thank you so much for listening and have a wonderful day.